Hey guys, for now we're gonna skip chapter 5. I know, right? It's like this is for the future anyways. I'm having struggle recording it. Unless somebody's out there like, dang it, he missed chapter 5 for now. Then who cares? So let's go right into DNA structure. So we need to know the difference between a nucleoside and a nucleotide. So nucleoside is when you only have your nitrogenous base plus your pentose, okay? Meaning these two. As you can see, I recorded this before, but you always have your nitrogenous base and your pentose sugar, okay? That's a nucleotide. That's a nucleoside. But when you add the phosphate, that's when you have a whole nucleotide, okay? So again, nitrogenous base and the sugar versus nitrogenous base, phosphate, and the sugar. And I would definitely note that difference for sure. You definitely need to know that DNA is formed 5 to 3 and 3 to 5, right? So we know that they're anti-parallel. And we need to go over your nitrogenous bases. So for the MCAT, you do need to know structure of all of these, and you also need to know a few important things. So you need to know T, thymine, only pairs with adenine, okay? And another thing you need to know is that they pair between 2 hydrogen bonds, okay? Thymine, adenine, 2 hydrogen bonds. Next, you need to know cysteine only pairs with guanine, okay? You need to know cysteine, guanine, and those make 3 hydrogen bonds. Okay, next we need to talk about uracil. Uracil is only found in RNA. It replaces thymine. So in RNA, your A doesn't bind to T, your A would bind to uracil, okay? So meaning we do not have thymine in RNA, okay? We have uracil. Uracil is only found in RNA, okay? And then we have pyrimidines and purines. So your purines are 2-membered rings, and then your pyrimidines are 1-membered, okay? So these two are your purines, and then your pyrimidines are going to be T, C, and U. Awesome. So next we're going to go to the Watson and Crick model, which is going to be this model over here. So this tells us that DNA is a right-hand helix, base pairing A to T, like we just said, 2 hydrogen bonds, and then G to C, 3 hydrogen bonds. And you do need to know that the G to C bond is going to be stronger than your A to T bond due to the extra hydrogen bond. Next, we need to talk about denaturing. So heat, pH, or chemicals denature DNA, but slow cooling and removal of the denatures allows DNA to coil up. So when we say denature, think of it like we're going to go from something that's clumped up to something that's like flat, right? And if you cool it, it gets clumped up again. Okay, so in chapter 6.2, we're going to talk about eukaryotic chromosome organization. So I will see you there.